Good afternoon, everyone. So as David mentioned, I work based at the Australian National Phenome Centre, ANPC, and we are quite blessed in terms of the type of analytical facilities that we have. So in today's presentation, what I'll do is to tell you a little bit about what I've done and how I utilize these advanced analytical chemistry instruments to give us better understanding of our product at a molecular level, and then how we can potentially use this information to add values to our products. And for this particular talk, the products that I refer to are these fresh produce. So the first question that I want to ask everyone is, can you name all of these fresh produce that I've shown on this slide? If you don't worry, because when I was first shown, I can only name 2 of them. So let's start by looking at the picture on the left-hand side. These are globe artichoke. And moving along to this looks like a potato, but not potato. And this is a yacon. And further to the right, something that looks like a ginger, but these are actually Jerusalem artichoke. And at the bottom, these are looks like overgrown garlic, but instead they are elephant garlic. And I've also put pictures of the common garlic that we commonly see, and just so you can see the size of that. So these are the produce that we will analyze in our project. And there's one particular bioactive component that is common to all these 4 different plant samples. And this particular bioactive component is, oops, I'm not sure why it's not moving, is something called inulin. And it is actually a dietary fiber. And inulin consists of a lot of different fructose units that are joined together by a beta-2,1 glycosidic bond, and it's end with a glucose unit. And usually the way how we refer to inulin, we talk about the degree of polymerization, or DP, and this refers to how many repeating fructose unit that we have in the inulin molecules. And the DP is actually affected by the type of plant species, as well as the way how we cultivate the plants itself, including the harvesting time. So why is the fascinations about this particular compound inulin? That's because of this particular beta-2,1 glycosidic bond that makes it very unique. The reason why I say that is because this particular dietary fiber, as we consume them, we are not able to digest it. It will pass through our upper gut until it gets to our lower gut, where it's being fermented by our gut bacteria. And this is what a lot of potential health benefits can emerge out from there. For example, maybe a lot of people had heard about inulin being a prebiotic, because that increased the kind of good bacteria in our gut, such as lactobacilli or the bifidobacteria. And these different type of bacteria in our gut also releases gases and different chemicals. And some of these chemicals, such as short-chain fatty acid, that also can cause a number of different potential health benefits, such as reducing the blood sugar and suppress the satiety or the appetite hormone, as well as regulate the blood lipid. In today's talk, I'm not going to talk about the mechanism behind that and also some of the potential health benefit, because there's a lot of literature available. But what we do know is that, and the degree of polymerization of inulin can impact in terms of how quickly it's being fermented and what are the type of chemical that's being released by the bacteria in our gut. So at the moment, when we analyze inulin, there's no particular gold standard methods that we can use. And currently, the AOSE method is typically used to measure the total sugar content in the product. And the number that we will give, that they tend to provide, is about the average DP. So it doesn't tell us anything about the distributions of the inulin, whether they are, what's the concentrations of each of those DP as well. Although there are newer methods that can be used to measure inulin, but some of these methods also have its limitation. For example, they have poor resolution or poor reproducibility. And one of the biggest challenge in terms of trying to understand inulin better is the ability to try to quantify each of those individual DP of inulin compound in there. This is because there's no reference material that will allow us to generate calibration curves and therefore to quantify them successfully. So what we set out to do is, can we develop a method that is quick and is also highly reproducible to characterize the inulin in the sample? And if we can characterize the different degree of polymerization in the sample, can we then quantify them? If we can quantify them, can we then also apply it to characterize and quantify all the 4 different samples that I have shown you before in the first slide? And to do this, we use one of the most expensive instruments that's actually available in the ANPC, and it is called magnetic resonance mass spec, MRMS here. I'm not going to go into detail in terms of the method development and how we can use to measure inulin, but this is just an overview of the methods and the pipeline that we use. It is actually quite simple in terms of the sample preparation. We freeze dry them and we perform hot water extraction, and then we perform purification using SPE column, and then we spit back it and concentrate the extract. And with the sample with the extract, we then mix it with a chemical matrix. And this is important because the chemical matrix can make the experiment being non-reproducible. So as part of this experiment, we compare 4 different types of matrix in order to identify the most suitable matrix. Once we have identified the suitable matrix, then we put it through for analysis, and then we can look at the data to see what sort of distributions of the inulin in the sample. So briefly, I'll show you a little bit of the results. So I've mentioned earlier on that the chemical matrix that we use is important. Here I show you 2 of the results. The one on the top, we can see that the solutions are very much homogeneous compared to another example that I've shown at the bottom, which is CHCA, that shows lots of crystallization. And these actually resulted really poor results. And for CHCA-BA matrix, we can actually see a very clear distinguish for the different type of DP in the inulin. Not only is very clear, the peak intensity is also high, and in comparison to all the 3 other matrix that we have used, the peak shape is good and the intensity is also much better compared to all of them. And because of that, we opt to use this CHCA-BA matrix. And the next step that we want to do is, can we then generate our result in a reproducible way? And also, I've mentioned earlier on that because we don't have a reference compounds to perform calibration curves, then how can we prevent that and still allow us from performing quantitative work? So the way how we go about it is to use a chicory inulin powder, and we mix it with the CHCA-BA matrix. And what we subsequently adding to do is that we increase the concentration of the chicory inulin powder and see what happened to the individual DP of inulin. Here I've shown you some of the results, and you can see that as we increase the concentration, each DP is also increased in a linear way. And we can see that the R-square value are typically very good, more than 0.9. And in addition to that, we also perform replicates of analysis, and we look at the reproducibility of each of the analyses between different samples. So we can see that the coefficients of variations is very good, which is less than 20%. And this is what's the FDA standard for developing a method. Here I've shown you the result up to DP20, but in fact, we can actually quantify the DP up to 25, and we can also detect the signal for DP that are much bigger. At the moment, based on the literature, we can see some of the methods is capable of detecting and quantifying up to DP13. So we think this particular approach is an approach that we can use to then analyze our plant sample. So here's the quick results of what we have done so far for these 4 different plants. From here on the left-hand side, which is a PCA score plots. And what you can see is that on the first component, you can see a change in terms of the concentration, and in particular, higher amount of the lower DP of inulin. And for the second component, we can, And for the second component, we can see increasing in the chain length. So from this overall of the summary result, we can see that these 4 different type of plant sample actually have very different concentration in terms of the different DP and also in terms of the chain length. And here I've shown you what we call it is a RKM plot, which is just a simplified versions of looking at the DP of inulin. So each dot here represents different DP of inulin, and with interval between each dot is a fructose unit. So you will know that as we go through a different dot, that's increasing 1 unit of fructose. So it's increasing in the chain length of the inulin. And the size of the dot is also corresponds to the relative abundance of the DP. So the bigger the dot, the higher the concentration that is. And you can see that for the 4 different type of plant sample, Yacon has the shortest range, which is up to 19, relatively similar chain length for Jerusalem artichoke and globe artichoke, and whereas elephant garlic, which has much longer chain length up to 45. And we also perform quantifications of the overall inulin content, and we found that Jerusalem artichoke has the highest concentration compared to all the other 3 compounds, whereas elephant garlic tends to have a much higher concentration on the longer chain length. So in summary, what I've shown you is the method that we have developed is highly reproducible and is very fast. We can analyze each sample in duplicates in less than 2 minutes. And I've also shown you that the result for this different type of plants, we have a number of, we have a very different distributions of inulin. So what can we do with this information? As I have mentioned earlier on, depending on the way how we harvest the plant sample, we can actually affect the concentrations of the inulin in the sample. So in a way, we can use this information to then maybe modify the cultivations process so that we can emphasizing in the increased concentrations of certain DP of inulin. And at the same time, now that we know the different DP of inulin in different plant sample, it's actually allowed us to understand how it can actually have an impact on our gut health as well. And in fact, we actually just recently obtained an ethics approval for a human child to look at the different type of fiber, inulin fiber in particular, as well as compared to different type of dietary fiber and how that affects our gut health. And if you're in Perth region, feel free to scan on the QR code and to find out more about the study. And if you want to participate, we are currently recruiting for volunteers at the moment. So this is kind of the next step for our project, is to look at, now we understand the degree of polymerizations of inulin, what's the potential impact on on human health, really. So finally, I just want to thank the teams that who's been involved in doing the work, including technical supply for technical support for using the instrument, as well as hands-on data analysis. And of course, there's Future Food System and Mount Lindesay for providing funding and the different type of plant sample that I use in this particular project. So that's all for my talk for this afternoon.